Alright, today we're going to do the resting membrane potential and action potential. So I'm going to try to do my PowerPoint and hopefully clear up anything that you might not have understood in lecture. So you've got this in Blackboard. So between my lecture and this video and the videos you got from the other instructors, hopefully you guys will know all about resting membrane potential and action potentials. Okay. So let's do a little review from what we did in class the other day. First, since we're talking about action potentials, we're basically talking about something going on in the nervous system, right? So we're talking about a neuron. This is our neuron. And here you have your cell body. Remember the soma, which is basically the main part of the cell. You've got your dendrites, which receive information. They're the receptive or the receiving end. You have your nucleus, you have your long axon, which is the sending in. It sends the impulse of the information out. You have your axon terminals. You have myelin sheath, which here, this is the myelin, the type of cell that does that. It's called a Schwann cell. You have the myelin or the insulation around the axon, and you have the gaps called the nodes of Ranvier. You have this bulge called the axon hillock, and that's where the action potential starts on a neuron. Action potential goes in this direction. So action potential always goes in one direction. So when we talk about an action potential, we've got to basically talk about how you generate that charge. How do you get that battery, that plus and minus on the inside and outside of a cell in order to get an action potential? So let's do a little bit of review from back in Unit 1. We've got ion channels, which are proteins. And there's two main types of channels. We have non-gated, which we can call leak channels. They're always open, okay? Things basically diffuse from an area of high concentration to low concentration. So things go down their concentration gradient. So we have more that diffuse down to where there's less. You have gated channels, okay? They're either ligand gated or voltage gated. If it's ligand gated, that means a neurotransmitter like acetylcholine has to bind. If it's voltage gated, that means there's got to be some kind of change in the membrane potential, some kind of a charge, some kind of a current has got to come along and open up that channel. So ligand gated and voltage gated channels. Again, these channels, when they're open, like ions will flow down their concentration from high to low. Either inside or outside the cell, whichever direction is high to low, they're going to flow from high to low down their concentration gradient through diffusion. All right, so just kind of give yourself a visual on what these look like. Here are the different types of gated channels. This one is a ligand gated, okay? So you see it's closed, nothing's happening, sodium, potassium, there's no movement. Once acetylcholine, in this case the neurotransmitter acetylcholine, binds, that causes a shape change. The channel opens and sodium and potassium can both flow. All right. If it's a voltage gated channel, this is a voltage gated sodium channel. You see the membrane potential. It's closed. Nothing's happening. No sodium is able to pass. The voltage changes. Okay. There's a shape change. The channel opens and sodium can then enter. So you have ligand gated or voltage gated channels. If you remember a little bit from class, this is what's going on in the neuron. All right. Whoops, wait a minute, wrong side. This is going on in the neuron. And this is what you're seeing on the muscle side. That acetylcholine going across the synapse, binding to the muscle and opening up those ligand-gated channels. Okay, so remember, first thing we've got to do, we have to establish that resting membrane potential. We've got to get that battery started. Remember, you can do this. You just take a voltmeter and you just stick the little probe inside the cell and you can measure the voltage. Resting membrane potential on neurons is about minus 70 millivolts. That means it's negative on the inside of the cell relative to the outside. The inside is more negative. And you get that differential in charge based on what's inside the cell and outside the cell. That ICF is intracellular fluid. ECF is extracellular fluid. So the difference in the amount of sodium, potassium, chloride, proteins, things with charge, there's more or less on the inside or outside the cell, and that differential builds up that charge. It's also based on how permeable the membrane is. So let's go back to our cell from Unit 1. Here's our cell. You remember there's more sodium on the outside of the cell in your extracellular fluid. There's more potassium on the inside. There's more chloride on the outside and proteins on the inside. Chloride and proteins are considered anions. They have a negative charge. Sodium and potassium are positive. They have a positive charge, or cations. All right, so somehow you would think if you looked at this, you go, oh, that's probably balanced. You got enough positive and negatives on both sides that should be balanced. Well, in a perfect world where you had no channels whatsoever, nothing, no movement of ions, then it would be neutral. It would be no charge. It would be a neutral charge. But because of a special setup of leak channels and something called a sodium-potassium ATPase, or a pump, we have a differential distribution of these ions. Let me show you how that happens. You basically have more potassium channels than you have sodium channels. So for every one sodium channel, you have about 75 potassium channels. Okay? That means a whole lot more potassium can flow out of the cell than sodium can flow into the cell. So every time potassium leaves, it takes a plus with it. So more pluses are leaving than pluses are entering. So that gives it a more negative charge on the inside. In addition, you have these large proteins. What's going on in a cell? It's making proteins, and proteins tend to have a negative charge. They're not permeable. They don't leave the cell. Okay, so that negative charge stays inside the cell. Chloride gives you your negative charge on the outside. It can actually cross back and forth, and because it's freely permeable, it doesn't really change the charge. You get your negative charge from the large number of proteins and a large amount of potassium that's leaving the cell. Now, if you just let potassium diffuse out of the cell, eventually it would become equal. You would have the same amount of potassium on the outside as you do the inside, but that's not the case. We have something special called the sodium-potassium ATPase. And it's basically our pump. You may see it called the sodium-potassium pump. That means it requires ATP or energy to pump an ion against its electrochemical or its chemical gradient. So what happens, the sodium-potassium ATPase uses ATP to pump potassium back into the cell and sodium out against their concentration gradient. So you're pumping in potassium, maintaining that high amount of potassium on the inside so that it leaves and it gives you that negative charge on the inside of the cell compared to the outside. So now we've set up our battery. All right, so let's just show you one more time kind of what that looks like. If you're kind of a visual person, you need to see what those channels look like. Here's your sodium and potassium. Remember, they're proteins. These are your leak channels. So potassium just leaks right on out of the cell. Just nothing stopping it. There's a hole. There it goes. You have more potassium than you have sodium. So sodium's coming in. More potassium is leaving. You add that ATPase, which is basically a protein that uses ATP. And it's kind of cool. It actually is like a shape change thing where ATP binds. It will actually grab potassium, grab sodium, and kind of open and close, like a little transport across the membrane. So instead of it flowing down, it actually picks it up, puts it in, and spits it out on the other side. So it uses energy, uses work to cause sodium and potassium to cross the membrane. Sodium's going that way. Potassium is going that way. Okay, so when you have a change in a membrane potential, when that resting membrane potential changes, that's when you get what's called an action potential. All right, so that's a signal or an impulse. All right, so membrane potentials will change when the concentration of the ions across the membrane changes. And that's either due to a change in the actual concentration or the permeability of the membrane. More channels are opening or less channels are opening, or more channels are closing, less channels are closing. Something's causing the permeability of the membrane to change, allowing sodium and potassium to move across the membrane. All right, now these changes in membrane potential, that's what we call an action potential. How do those occur? All right, this is basically, we did this in class. This is a picture of an action potential. You have time, you have voltage. Remember, you're just measuring the voltage right inside the cell. And it's more negative inside than it is positive. Positive on the outside, negative on the inside. At rest, it's minus 70 millivolts. So nothing's happening. It's just going along at minus 70 millivolts. Some kind of stimulus comes along. That stimulus opens voltage-gated sodium channels. When you open those voltage-gated sodium channels, that allows sodium to flow into the cell with its positive charges. The more positive charges are flowing into the cell causes the membrane, causes it to become more positive on the inside. So you're seeing a change toward more positive the membrane potential. That's depolarization. Once you get to about positive 30 millivolts, those sodium channels will close, those voltage-gated sodium channels will close, and voltage-gated potassium channels will open. So those voltage-gated potassium channels open, the sodium channels close, potassium can then exit the membrane. Okay? More positives going out. As those positives leave, the membrane potential comes back down toward resting, and that's called repolarization. These voltage-gated potassium channels will stay open a little bit too long, causing the membrane potential to go below minus 70 before it comes back to rest. And this area here is called hyperpolarization. So it goes a little bit negative before it returns to resting membrane potential. All right? Now you'll notice something here called threshold. I didn't touch on this in class yet. I was going to do this later. Something called threshold. Action potentials, once they go, they go. Once an action potential reaches minus 55, it's going to go. So let's pretend that you sent a small signal, just a little bit of a stimulus came, and a few voltage-gated sodium channels opened, but not much. It would just stop. The action potential would not occur. You've got to reach that threshold. You have to have enough sodium channels open to reach that threshold, then the action potential will go. There aren't any that are this tall or this tall or this tall or that tall. Once you get an action potential, it's always the same. So they're all or none. All right, generation of action potential. At rest, all you've got going on at rest, remember this is at minus 70 millivolts, just sodium and potassium, just basically going across the membrane, setting up that resting membrane potential. Nothing going on. All your voltage gated channels are closed at rest. Once you start an action potential, your sodium channels, your voltage gated sodium channels, you have two. You have those that are closed at rest, that open with depolarization, which are activation gates, and then inactivation gates. They're open at rest, and they block the channel. So on your protein, basically have two gates, an activation gate and an inactivation gate. Basically just think of it opens and closes, allows sodium to move. Potassium channels only have one gate. They're closed at rest, and they will open slowly upon depolarization. So when you get to the top of depolarization, they're all open. You quickly get repolarization. Depolarization occurs by opening the voltage-gated sodium channels, causing sodium to flow in. So depolarization, remember, is the inflow of sodium, causing you to get more positive, causing your membrane potential to go up toward positive. Repolarization, those sodium channels close. Okay, no more sodium can cross. The voltage-gated potassium channels open. Potassium leaves the cell. Those positives start to leave the cell. That brings the membrane potential back down to negative. Hyperpolarization, for review, that's when you kind of overshoot. Those potassium channels stay open a little bit too long, It causes an after hyperpolarization, like a little overshoot or undershoot. Takes it too low, and it comes back up. Now this hyperpolarization, this is important when we talk about propagation of an action potential. Because this membrane potential is so low, you can't activate those sodium channels. So those voltage-gated sodium channels, they won't activate. It's just too low. That keeps an action potential from going backwards. It won't go in the other direction. It'll only go one way down an axon. All right, so this is kind of broken down action potential to show you the movement of the different ions. Here's your action potential, the pink. Okay, you'll see during depolarization, there's your sodium coming in. Sodium permeability is going up. You reach the top of depolarization, your sodium permeability starts to come back down. At the top of the action potential, you start to see potassium permeability changing. So potassium is now doing what? Leaving the cell. And then its permeability comes back down. So you have sodium as part of depolarization, sodium permeability responsible for depolarization, potassium permeability for repolarization. All right. Now, once you're done, you've got to have a way to get back to rest. You've got to kind of reload the system. So the way you reload the system, you use the sodium-potassium pump or the sodium-potassium ATPase. And it basically helps to redistribute the ions back to the resting condition so you're ready to go again. Remember, that uses energy to pump sodium and potassium against their concentration gradients. It pumps potassium into the cell, sodium out of the cell. So when we want to propagate or send an action potential down an axon, remember, it's just basically a little wave of action potentials moving down an axon. So what you're seeing is a reversal of charges all the way down. So here it was negative, now it's positive. Now these are going to become positive, these are going to become negative, these will be positive, negative, positive, negative, positive, negative. It'll just keep going all the way down the axon as that action potential moves down all the way from the axon hillock to the axon terminals. If you will watch those videos, I promise you, on my RMP plays, the ones I've posted in Blackboard, it will help you kind of visualize that and see that happen. So make sure you spend some time watching that video, because for me, I'm a visual learner on some things, and so seeing those animations kind of helps me get that in my head to see in 3D what that looks like. Let's go back to that threshold. Again, if you have a weak depolarization, if maybe the stimulus is not strong enough, nothing will happen. You won't get an action potential. And this is important because your nervous system is electrically active. There's always some kind of electricity, a little bit of something going on all the time. And you wouldn't necessarily want to respond to something that wasn't true. This kind of is what happens with epileptics. They send out signals that are not quite true signals, and their body responds in kind of an odd way. They like seizures and things like that. And so you kind of have a break point, a threshold stimulus that you've got to get to that point in order for your body to respond. And so the threshold stimulus is something that's just strong enough to start that action potential. Now once it starts, it's all or nothing. You can't have a bigger action potential, okay? Once you have one, you have one. So if you have a sub-threshold stimulus, not enough to do anything, it won't go, you won't get a response. If you have a huge stimulus, doesn't matter, you still get the same amount of stimuli. You still get the same response. All right, so one more time, we're going to do this picture one more time. So here's your membrane potential, resting, minus 70 millivolts. You get a stimulus. Something causes a little bit, something causes a voltage change toward positive. Okay, and we'll talk about what these stimulus are when we get to Unit 4. I'm trying not to overwhelm you too much right now. But you're getting a stimulus, okay, that causes that membrane potential to start to go toward positive. We have a threshold stimulus. It's enough. Plenty of sodium channels, voltage-gated sodium channels begin to open. Sodium starts to flow into the cell. Becomes more and more and more positive. The membrane depolarizes. It reaches about positive 30. Those voltage-gated sodium channels will close. No more sodium will enter. Voltage-gated potassium channels will open, and potassium will then exit the cell, along with all its positive charges. bringing the membrane potential back down to negative. It will overshoot just a little bit, so potassium ATPase will kick in, and we will return to our resting membrane potential. Okay, so I hope that answers some of your questions. I know it's a complicated part of what we're learning right now, but between my lecture and this video and the videos that you've got from the other instructors and from your My MP Place videos, hopefully you guys will get it. Spend some time on it. There's some really good questions at the end of your chapter. If you want to look at that, make sure you can answer those questions. Ask me questions. Do your quizzes. And we'll keep at this. We're going to see this for the next week, and then we'll do it again when we get to the nervous system. So hang in there and come ask me if you have questions, and I will see you later. Thanks.